Good day, dear students. Welcome to this topic on DNA replication. The purpose of DNA replication is to produce two identical copies of a DNA molecule. This is important for cell division during growth or repair of damaged tissues. DNA replication, therefore, ensures that each new cell receives its own copy of the DNA. So before I explain to you the process of how DNA molecules are copied, let me first present to you the alternative models of DNA replication. These models of DNA replication emerged immediately after Watson and Crick successfully identified and created a structural model of the DNA molecule, which is the double helix. The first proposed model that I will talk about is the conservative model. Please refer to the picture provided. The dark blue strands are the parental or the original DNA strands, while the light blue ones are the new strands. In this model of DNA replication, the two parental strands are said to be reassociated or reattached after acting as templates for new strands, thus restoring the parental double helix. Meanwhile, the semi-conservative model is characterized as having the two strands of the parental molecule separating, thereby functioning as templates for the synthesis of new complementary strands. This was also the same model supported by Watson and Crick. Lastly, the dispersive model. In this model, each strand of both daughter molecules contains a mixture of old and newly synthesized DNA, thus the term dispersive. With the three emerging models of DNA replication, Matthew Meselson and Franklin Stahl devised a clever experiment that distinguished between the three models. The results of their experiment supported the semi-conservative model of DNA replication as predicted by Watson and Crick. The basic principle of DNA replication is conceptually simple. However, the actual process involves some complicated biochemical gymnastics, as we will now see. So now let us look into the initial process of how DNA is replicated. DNA replication commences at the origins of replication. The origins of replication contain short stretches of DNA having a specific nucleotide sequence. Special proteins recognize the specific nucleotide sequence and attach to the DNA. As a result, the two strands separate and a replication bubble forms. Then, replication proceeds in both directions until the entire molecule is copied. Here is an example of the origin of replication of a bacterial chromosome, specifically that of E. coli. The E. coli chromosome, like many other bacterial chromosomes, is circular and has a single origin. Proteins that initiate DNA replication recognize this sequence and attach to the DNA, separating the two strands and opening up a replication bubble. In contrast to a bacterial chromosome, a eukaryotic chromosome may have hundreds to even a few thousand replication origins. In this example, multiple replication bubbles form and eventually fuse, thus speeding up the copying of the very long DNA molecules. Similar to the replication of the bacterial DNA, eukaryotic DNA replication proceeds in both directions from each origin. Then, from the ends of the replication bubbles, a replication fork forms. This is a Y-shaped region where the parental DNA strands are being unwound. Several kinds of proteins participate in the unwinding of the double helix. Helicases are enzymes that untwist the double helix at the replication forks, separating the two parental strands and making them available as template strands. After the parental strands separate, single-strand binding proteins bind to the unpaired DNA strands, keeping them from repairing. The untwisting of the double helix causes tighter twisting and strain ahead of the replication fork. Topoisomerase helps relieve this strain by breaking, swiveling, and rejoining DNA strands. The initial nucleotide chain that is produced during DNA synthesis is actually a short stretch of RNA and not DNA. This RNA chain is called a primer and is synthesized by the enzyme primase. Primase starts a complementary RNA chain from a single RNA nucleotide, adding RNA nucleotides one at a time, using the parental DNA strand as a template. We have this figure to summarize the different proteins involved in the initiation of DNA replication. Helicase unwinds and separates the parental DNA strands. Single-strand binding proteins stabilize the unwound parental strands. Topoisomerase breaks, swivels, and rejoins the parental DNA ahead of the replication fork, relieving the strain caused by unwinding. And lastly, primase, which synthesizes RNA primers using the parental DNA as template. Now that you have learned about how DNA molecules unwind, it is time for us to understand how a new strand of DNA is synthesized and formed. Vital enzymes in the synthesis of a new DNA strand are DNA polymerases. DNA polymerases are enzymes that create DNA molecules by assembling nucleotides or the building blocks of DNA. These enzymes are essential to DNA replication and usually work in pairs to create two identical DNA strands from one original DNA molecule. These enzymes work by adding nucleotides to a pre-existing chain in the presence of a primer, which is actually a short stretch of RNA. The rate of elongation is about 500 nucleotides in bacteria and 50 per second in human cells. Each nucleotide added to a growing DNA strand comes from a nucleoside triphosphate, which is a nucleoside or a sugar and a base with 3 phosphate groups. The nucleoside triphosphates used for DNA synthesis are chemically reactive, partly because their triphosphate tails have an unstable cluster of negative charge. DNA polymerase catalyzes the addition of a nucleoside triphosphate to the 3' end of a growing DNA strand with a release of 2 phosphates. The 2 ends of a DNA strand are different, giving each strand directionality like a one-way street. In addition, the 2 strands of DNA in a double helix are antiparallel, meaning that they are oriented in opposite directions to each other like a divided highway. Clearly, the 2 new strands formed during DNA replication must also be antiparallel to their template strands. In the antiparallel elongation of the DNA strand, DNA polymerases add nucleotides only to the free 3' end of a growing strand. Therefore, a new DNA strand can only elongate in the 5' to 3' direction. Along one template strand of DNA, the DNA polymerase synthesizes a leading strand continuously, moving toward the replication fork. This figure shows the synthesis of the leading strand during DNA replication. After the RNA primer is made by the enzyme primase, DNA polymerase starts to synthesize the leading strand. Furthermore, the leading strand is elongated continuously in the 5' to 3' direction as the fork progresses. Again, in the synthesis of the leading strand, there is continuous DNA synthesis toward the direction of the replication fork. Another strand, the lagging strand, is elongated almost at the same time the leading strand is made. To elongate the other strand called the lagging strand, DNA polymerase must work in the direction opposite or away from the replication fork. The lagging strand is synthesized as a series of segments called Okazaki fragments, and these fragments are glued or joined together by DNA ligase. This figure shows the overview of the entire DNA replication process. As mentioned, DNA replication starts at the origins of replication, where helicase unwinds the parental double helix. Next, the unwound template strands are prevented from repairing by the single-strand binding protein molecules. Then, there is continuous synthesis of the leading strand in the 5' to 3' direction by DNA polymerase. In the photo, The lagging strand below is synthesized as a series of 5 Okazaki fragments. The figure also shows the enzyme primase beginning to synthesize the primer for the 5th Okazaki fragment. Depicted also in the picture is the behavior of DNA polymerase after the synthesis of an Okazaki fragment is completed. The gluing enzyme DNA ligase joins the ends of the Okazaki fragments together, completing the entire length of the lagging strand. The proteins that participate in DNA replication form a large complex, a DNA replication machine. The DNA replication machine may be stationary during the replication process. Also, recent studies support a model in which DNA polymerase molecules reel in parental DNA and extrude newly made daughter DNA molecules. Primase may act as a molecular brake, coordinating primer placement and the rates of replication on the leading and lagging strands. This photo is a current model of the DNA replication complex. Two DNA polymerase III molecules work together in a complex, one on each template strand. The lagging strand template DNA loops through the complex. DNA replication is not 100% accurate. That is why, during DNA replication, DNA polymerases proofread each nucleotide against its template as soon as it is added to the growing strand. Upon finding an incorrectly paired nucleotide, the polymerase removes the nucleotide and then resumes synthesis. This action is similar to fixing a typing error by deleting the wrong letter and then entering the correct letter. In mismatch repair of DNA, repair enzymes correct errors in base pairing. In addition, DNA can also be damaged by exposure to harmful chemical or physical agents such as cigarette smoke and X-rays. It can also undergo spontaneous changes. In nucleotide excision repair, a nuclease cuts out and replaces damaged stretches of DNA from harmful chemical exposure. This figure shows how DNA damage is repaired through nucleotide excision repair. First, teams of enzymes detect and repair damaged DNA, in this case this thymine dimer which is caused by ultraviolet radiation, which distorts the DNA molecule. Next, a nuclease enzyme cuts the damaged DNA strand at 2 points and the damaged section is removed. Then, repair synthesis by a DNA polymerase fills in the missing nucleotides, and lastly, DNA ligase seals the free end of the new DNA to the old DNA, making the strand complete. Now let us move on to the next part of my discussion, which is about replicating the ends of the DNA molecules. In spite of the impressive capabilities of DNA polymerases, there is a small portion of the cell's DNA that DNA polymerases can neither replicate nor repair. Limitations of DNA polymerases create problems for the linear DNA of eukaryotic chromosomes. The usual replication machinery provides no way to complete the 5' ends, so repeated rounds of replication produce shorter DNA molecules with uneven or staggered ends. Most prokaryotes have a circular chromosome with no ends, so the shortening of DNA does not occur. But what protects the genes of linear eukaryotic chromosomes from being eroded away during successive rounds of DNA replication? It turns out that eukaryotic chromosomal DNA have special nucleotide sequences called telomeres at their ends. Telomeres do not contain genes. Instead, the DNA typically consists of multiple repetitions of one short nucleotide sequence. Telomeres do not prevent the shortening of DNA molecules, but they do postpone the erosion of genes near the ends of DNA molecules. It has been proposed that the shortening of telomeres is connected to aging. Telomeric DNA acts as a kind of buffer zone that protects the organism's genes. In addition, specific proteins associated with telomeric DNA prevent the staggered ends of the daughter molecule from activating the cell systems for monitoring DNA damage. If chromosomes of germ cells become shorter in every cell cycle, essential genes would eventually be missing from the gametes they produce. That is why an enzyme telomerase catalyzes the lengthening of telomeres in germ cells. Telomere shortening might protect cells from cancerous growth by limiting the number of cell division. However, research also suggests that there is evidence of telomerase activity in cancer cells, which may allow cancer cells to persist. So that's it for my discussion on DNA replication. I hope you have learned something. Thank you for watching this video.